Okay, we're going to look at paper 3. Okay, I only mark the beginning of paper 3 lah. Okay, so for the first question, right, the question says with reference to figure 1.1 and 1.2, right? That means the answers to both part 1 and 2 should come from the information given in the two figures. Okay, so to suggest, right, how the thyroid hormones are able to act on nearly every cell in the body, right? Okay, let me look at the kind of like a wrong answer first. Yeah, so many of you suggested this, right? Many of you suggested the presence of receptors that the thyroid hormones is specific to in all the cells. All right. While this may be true, we don't know, right? Or at least so far in this question, right? You don't know yet. But the presence of such receptors is not shown, right, in figure 1.1 and 1.2. Right. So even though this is logically correct, right, it is not the answer, right? So in figure 1.1, we see that the thyroid gland, right, and the information mentioned, the thyroid gland is lined with thyroid follicular cells, right? Okay, so the thyroid gland with the thyroid follicular cells, we can see in figure 1.1 that there is the blood capillaries very near, right, to the cells. Okay, so then this should therefore be the reason, according to figure 1.1, how the thyroid hormones are able to be transported to all cells in the body and then somehow, right, act on all cells in the body, right, restricted to the information given in figure 1.1 and 1.2. Some of you use figure 1.2, right, and mention that, oh, because there are all these different functional groups, right, in the thyroid hormones, and somehow they are able to bind to multiple receptors. So again, the receptors that the thyroid glands can bind to is not shown, right, in figure 1.1 or 1.2. Anyway, such thinking is flawed, right? Because if you think about an insulin, a glucagon protein hormone, they also have a lot of R groups, right? A lot of different functional groups, right? But they don't bind to a wide range of receptors, right? So just having different functional groups doesn't mean you can bind to different receptors, right? You should still have a receptor that is overall of that has a binding site that is overall of a complementary shape, right, to the shape of the thyroid hormone. Okay, so for those who have written that presence of different functional groups according to figure 1.2 and somehow being able to bind to different receptors found on all cells, that is a bit flawed in the logic. Okay, so anyway, back to this. Okay, so restricted to the information in figure 1.1, the answer would be the presence of the capillaries, which allows the transport of the thyroid hormones throughout the body, right, via the bloodstream. I thought this would be the most complete answer because some of you just mentioned the capillaries, full stop. Some of you didn't mention the capillaries and just say that the thyroid hormones can be transported via blood. I've given it to you, but the most complete answer is you need to mention the capillaries, which you can see, right, in figure 1.1. So this would be the quoting of information, and then the reasoning, which is the ability to be transported throughout the body via the bloodstream. Okay, then for part 2, right, the compare, right? So both similarities and differences. Okay, 2 marks, so 1 similarity, 1 difference. So compare between the structure of thyroid hormones and amino acid. So the thyroid hormone, I've rearranged the thyroid hormone to look like the normal configuration of an amino acid. Okay, you have the central carbon, same as the amino acid. There is the amino group, same as amino acid. There's the carboxyl group, same as the amino acid. Hydrogen atom, same as the amino acid. Amino acid has a R group, right, for the thyroid hormone. It's not called an R group, but structurally you can see that it is similar in terms of the bonding to the alpha, the central carbon, right? So Therefore, this would be the similarity with the amino acid, that both the thyroid hormone and amino acid has a similar general structure. Okay, the difference is in the R group equivalent. There are iodine atoms, whereby we know that for amino acids there are no iodines, right? Such an atom is not found in any of the amino acids. So that would be the most obvious, the most important difference. So you try to compare the structure of the R group. So you are not expected to know the structures of all the R groups, right? So if you are not expected to know, and seriously you don't know, right, then why use such or why try to compare, right, with the structure of the R group? And I thought the presence of the iodine are very obvious. All right, going back to figure 1.2, right, some of you mentioned, you have the amino group here, the carboxyl group here, and they are found on one side. Versus in amino acid, you have amino group on the left and carboxyl group on the right. That is really the orientation, how you see the molecule, right? So again, if you flip it and look at it this way, then you have the amino group on the left, carboxyl group on the right. So that is the placement of the amino group and carboxyl group found on one end, not on opposite end. That is really not a difference. Okay, a bit more feedback, right? The name is amino group, right? Not amine, not amide. A lot of you put amine. I suppose that's what you have learned in chemistry. Okay, the carboxyl group. So a lot of you mention it as the carboxylic acid group, but it is a carboxylic acid, but you don't call it a carboxylic acid group. It's called carboxyl group, right? And certainly not carbon now. Okay. So please name the functional groups of the amino acids correctly. Then some of you use N and C terminus. That is only applicable in a polypeptide chain, right? Terminus, right? So you have a chain, then you have two ends, right? And amino acid don't have ends, right? It's not a chain. Okay, so it's just two functional groups, amino group and carboxyl group. Okay. So this one mentioned already that the most obvious difference is the presence and absence of iodine. Yeah, that is applicable to all the amino acids. Okay, then move on to part B. A lot of information given. That is how paper 3 is like. You have a lot of information. You need to figure out what is important, what is not, right? And sometimes the information needed to answer a question might be quite far away, meaning you might have a question at the more at the back of, say, question 1, maybe like one part G, for example, but then the information is actually given right in front. Right? So there's a bit of reading, remembering what you read, right? Consolidating needed for paper 3. Okay, so that's why you have such large amount of information for you to experience that. Okay, then focusing on the graph, right? Compare again, right? So similarities and differences. Again, 2 marks, one similarity, one difference. Quite a number of you forgot the similarity, yeah? Okay, so for similarity, not much, right? The only similarity is that both decreases. Some of you focus on between day 5 and 6 and mention that they both decrease to the same gradient, which is a low gradient. That is acceptable as well, right? But I gave the overall as a overall decrease as a similarity. Okay, so for the difference, okay, take note of the units given, right? Some of you didn't even see this, right? You just mention level instead of using the unit, or you say arbitrary units. The exact units are given. Okay, and the T4 and T3, right, has different units. Right, T4 is in microgram, while T3 is in T4 is in milligram and T3 is in microgram. Right, milligram mg is 1,000 times bigger than microgram. Okay, so you need to take note of that. And quite a number of you mix up the two graphs, and obviously if you look at the shape of the graph, because mine has color, yours don't have, right? But you can still see the shape of the points, right? clearly the triangular points are is referring to T3, and this squarish ones is for T4, right? So a lot will mix up between the two. Okay, so for the differences, okay, the starting for T3 is actually lower, even though it looks higher on the graph, but because of the unit, right, microgram, so actually the starting level is lower than T4. The rate of decrease, depending on how you look at it, the rate of decrease in terms of gradient is higher for T3, but if you look at the absolute amount, right, then T4 actually decreases faster because of the, again, the unit microgram, right, decreasing by about 2, right, less than 2 microgram is still larger than a decrease of about 7 microgram, right? So again, decrease of 2 milligram. Okay, mixing up. Decrease of about 2 milligram is higher than a decrease of about 7 microgram. So really depends on in terms of what that you are talking about, whether T3 is faster or T4 is faster. So in the description of the difference, right, when you say T3 is faster, for example, you should actually go on to say faster in terms of maybe the gradient or similar words like that, right? Or for those who say T4 decreases faster in terms of the absolute amount, for example, right? But most of you did not actually explain what you are really comparing when you say one is faster than the other. I have given it to you, right? So this graph is made more complex because of a difference in the unit, right? So if such thing is given in the A level, you open your eyes and interpret the graph accordingly, right, with the different units. Okay, then for part 2, okay, it's still with reference to the figure, with reference to figure 1.4. All right, the differences, contrast between the release of the thyroid hormones and the release of antibodies, right? So two possible answers. The release of the thyroid hormone is on the same day when the TSH was introduced. If you read back on the information in the previous page, right, TSH is one of the hormones that trigger the release of thyroid hormones, right? So TSH is a trigger. Okay, so the day that TSH is introduced, straight away you have high level of thyroid hormones, meaning that the triggering of the release of the thyroid hormones is almost immediate, but at least on the same day, right? But you know that for antibodies, there is a lag time of a few days for the activation of the adaptive immune system, right? For the B cells to change to plasma cells to make the antibodies, right? So immediate versus lag time. Or of course, right, you can talk about what is triggering the release, right? So the trigger is by TSH for the thyroid hormones as shown in figure 1.5, 1.4 rather, right? And for the antibodies, the trigger is by cytokines from the helper T cells. Okay, make sure the contrast is of the correct feature. Yeah, so here we are talking about a chemical, a molecule that trigger, right, the release of your thyroid hormones versus antibodies. Some of you talk about triggered by TSH versus for antibodies, you talk about the cells that release the antibodies, plasma cells, right? So a molecule that trigger release compared to the cell that releases, this is not of the uniform or the same feature, right, of comparison. Right, so that is not accepted. So here, clear feature of comparison, we are talking about the molecule that is releasing, or rather the molecule that is triggering the release of either your thyroid hormones or antibodies. Okay. Again, so with reference to figure 1.4, so anything that is not shown in figure 1.4 shouldn't be in the answer, so, which means for many of you who mention where the thyroid hormones is released from, that is not found in figure 1.4. I know you have the information earlier, right? But restricted to information in figure 1.4, that is not what is shown. Okay, so saying that the thyroid hormones is released from the thyroid gland versus antibodies from the plasma cells, that is, while correct, not an acceptable answer here. Okay, move on to part C. Okay, part C, looking at the diagram, A is our adenyl cyclase because it converts ATP to CMP, right? And B, that is bound to GTP. B would be the G protein, right? Or activated G protein, as some of you have mentioned. That is very good. Okay, for a few of you who mentioned that B is the Ras protein, okay, so please do look at the comments here. While Ras protein is a G protein, Ras protein is specifically the G protein that is involved in the signaling pathway for cell growth, for cell division. Right, that's why when we have a mutated Ras, when the Ras protein is hyperactive, right, it causes or it contributes to cancer, right? Here, the context is not cell growth, right? Here, the context is the production and release of your thyroid hormones, right? So it's not specifically Ras protein, right? So it's just a G protein. We don't know what is the G protein that is specifically involved here. All we know is it's called G protein, right? So then just G protein rather than something more specific for something else, right? So the Ras protein is more specific, but for cell growth, right? Cell division. Then for again the few of you who mentioned that B is the receptor, you mentioned G protein linked receptor, right? The receptor is labeled as TSH receptor, right? So specifically for B, that is just a G protein. Okay, that's why you lost mark there. Most of you got A correct, only the very few of you got A wrong. That one is really a lack of revision, not familiar, I suppose. Okay, then for part 2, right, the significance of protein A. Okay, so protein A is the adenyl cyclase, right, as you have identified earlier. So it functions to convert the ATP to CMP, right? And then we can talk about what CMP does, right, as part of the significance of protein A, right? So what's the significance of converting ATP to CMP, right? So CMP will activate pKa. But however, right, most of you just talk about CMP binds to pKa, conformation change, then activate. That is not what is shown in figure 1.5, right? So when more information is given, you need to base it on the information. So clearly here you can see that the CMP, these are the CMP for those who cannot interpret the diagram, right? These are the CMP. So the CMP actually binds to not the pKa, right? It binds to the R. Okay, so call it R dimer, call it repressor R, whatever you want to call it, right? R, right? So it binds to R to release the pKa, right? So that activation process, which is shown in more details in this diagram, should be described rather than just a simple conformation change and activation. Okay, so that's why most of you don't get your second point, which is for the activation of pKa. Okay, then the third point, the active pKa. Okay, most of you went on to talk about how it triggers the phosphorylation cascade, but you just say it triggers phosphorylation cascade, right? You didn't say how, right? So pKa is a kinase, right? Protein kinase A. So kinase catalyzes phosphorylation of other proteins, right? Or other kinases. So in this case, the pKa would phosphorylate another kinase, which this kinase then get activated and phosphorylate another kinase, and so on and so forth. And that is the phosphorylation cascade, right? So to trigger the phosphorylation cascade, just a bit more details to talk about how pKa activates another kinase via phosphorylation. Okay, then lastly, right, for the signal amplification, we are focused on protein A. So we are not talking about signal amplification by pKa during the phosphorylation cascade. We are talking about signal amplification by protein A, all right? So specifically for protein A, during the conversion of ATP to CMP, there are many CMP molecules produced, right? So that then allows for signal amplification. Okay, many. So the word many, or the idea that there are multiple CMP molecules produced by one adenyl cyclase, right, should be clear, right, in your answer in order to score this fourth point. All right. The comments I've mentioned just now as part of the explanation, so I'll leave you to read it further. Okay, I'm just going to move on to part 3, right? This one also according to the figure, you can see that there's an organelle that is releasing right, the calcium ions, right? So back to the figure 1.5. Look at this organelle. From this structure, you need to identify what is this organelle, right? So this organelle has got flattened cisternae. You can see that the cisternae are joined together, right? They are not independent layers of cisternae, right? Unlike Golgi. Yeah, they are interconnected, as you can see here very clearly, this part especially. Yeah, so interconnected cisternae cannot be Golgi apparatus, which many of you mentioned. There are no ribosomes, right, on this membrane of this organelle, so it cannot be rough ER. So really, from the structure, all the organelles should be the smooth ER, right? Also, if you are familiar with the functions of the different organelles, storage of calcium ions is one of the function of this smooth ER. Okay, so this question not only tests the signaling, right? Not only does it test your interpretation of the diagram, also testing you on your organelles, right? Which, well, quite a large majority of you couldn't figure out that this is the smooth ER. Okay, so identify that this is a smooth ER, then the question asks for structures of this organelle, right? So you got to mention the respective structure that allows for the function, right, during TSH signaling. So one of the structure is that it is membrane bound and that the membrane is impermeable to calcium ions due to the presence of the hydrophobic core. So this allows for storage, right, that the calcium ions don't leak out of the organelle. And then, right, there are the presence of IP3 gated calcium channels. The name is written in figure 1.5, right? IP3 gated calcium channels. Right, so when you are naming this structure, then you should follow what is given, right, in the information. So for those who just mentioned channel, calcium channel, right, those I did not credit you, you should cite the full name as given, right? IP3 gated calcium channels. So this allow for the facilitated diffusion of calcium out of the SCR into the cytoplasm when IP3 binds to these channels, right? So IP3 gated means IP3 binding opens the channel, right? So when IP3 binds, the channels are open. That allows the calcium to leave, right? The smooth endoplasmic reticulum into the cytoplasm. Some of you mentioned the cytoplasm as what intracellular space. Just because it's labeled intracellular here, this is just telling you that this is inside the cell. What is inside the cell here? Cytoplasm, right? So we've got such thing as intracellular space, right? So cytoplasm, yeah. Then we can talk about the function of calcium itself, right? So what is the importance in releasing the calcium in terms of TSH signaling? That is to bind to and activate the calcium-dependent protein kinase as given in the diagram, right? The full name. That will go on to further propagate the signal, right, in the signal transduction pathway. Then back to the structure of the smooth ER, you can mention the large surface area, right? But between large surface area and efficient release, you should mention the insertion of more IP3 IP3 gated calcium channels, right? So that is the logical explanation, yeah, or description. Large surface area so that there are more channels, so that there is more efficient release of calcium. All right. So again, most of the feedback comments I have put into the explanation just now. Right? I'll leave you to read it further lah. Okay, so these are the questions I've marked for paper 3.